Now share my PowerPoint. Share. Can you see my slides now?
Yes, you need to go into the full screen mode. Yeah.
I'll do that. You see if I can imitate. Spotlight. Okay, can you see my cursor? Hello?
No,
you are kind of stuck on the slideshow. Your mouse is stuck there for us.
you need to go.
Okay, let's see. Do you see it now?
No, I don't at least. So you do, yeah.
Yeah, it's okay.
Yeah.
Yes, we can see it now.
Yeah, it's
Good.
I want to show slideshow.
Yeah, now just go to the full screen and then you're ready to go.
It
should be something on the bottom towards the right. It should say, there's an icon for full screen usually.
No, I just want to move. Okay, let's see. Want to be able to you guys to see my. Can you see my cursor now?
Yeah, no, yes, we could see your cursor. I mean, it kind of disappeared, but we could see it for a while.
Okay, so let me just get started then.
Yeah, so all right, fine, okay, you can get started. I mean, there should be an icon that you could just press to get into full screen mode so that you don't have the set of slides. But don't worry about it, just go with what you have.
It's in full screen now. I'm in full screen.
Not for us, Ghanim. We don't see in full screen. Now we can see it. Now we can see.
All right, good. Go.
Okay. So actually, just hi everyone. Those just to introduce myself, I'm Ghanim Ullah from University of South Florida. I'm an associate professor here. This is where I am, Tampa, in Florida, just 50 miles from where Mickey Mouse and Donald Duck live. And so University of South Florida is in the northeast of Tampa, a very nice area, and this is Department of Physics and Molecular Biology. So my lab is in this building. So first up, the contributions. This work is the experimental data comes from a longtime collaborator, Angelo Timuro, who is at University of California, Irvine. And the modeling is done by Dr. Sayed Shah, who is a postdoc in the lab, and Dr. Patrick Toglia, who was a graduate student and got his PhD a couple of years ago. And the funding comes from the National Institute of Health. So just to quickly introduce I mean, I'm sure you guys have heard of Alzheimer's disease. It's it's a devastating aging and genetic type of disease where it starts with just the degeneration of synapses and then memory loss and eventually dementia and eventually death. The long-standing hypothesis in the field is just a cartoon version of it, is that there are these proteins called amyloid beta, and normally brain produce and clear these proteins, but something happens, something goes wrong where the brain either produces more amyloid beta or it's unable to clean it properly. And what that does, that these amyloid beta, they start making aggregates. So multiple proteins, monomers, they come together and they take these oligomeric shapes. And they first start accumulating inside the cell, but then they are released into the extracellular space. And there is usually a cycle going on, these proteins being released and they go inside and come back. In all this process, they do a lot of bad things to the cell. So the chain of events that happen in genetic type of Alzheimer's disease, for example, there are mutations in these 3 different types of proteins, amyloid precursor protein, presenidine 1 and 2, that would lead to the increased production of amyloid beta, which leads to the aggregation of amyloid beta, and amyloid beta 42, the 42 amino acid type of amyloid beta is the most toxic one. So, but there are others like amyloid 40, 41, and 43. They all have some level of toxicity. That leads to the first targets are the synapses. So these oligomers, they first target synapses, kind of just leads to their dysfunction. That also leads to microglial and astrocytic activation, which is also bad. It causes neuroinflammation in the brain and the release of cytokines, which are which kind of it enhances, exaggerated this damage in the brain, and then eventually synaptic and neuronal injury, altered neuronal ionic homeostasis, like calcium signaling, for example, oxidative injury, and then large-scale tangles formation, and eventually dementia. So this is kind of a linear theory of Alzheimer's disease, but there is, and it's a very long-standing hypothesis in the field. But there are 2 problems with it. One of the problems is actually it turns out now that, and there is significant data, that there is no correlation between amyloid beta deposits and disease status in postmortem brains. So you basically, you will find brains of patients with Alzheimer's disease where they would have really severe Alzheimer's disease symptoms, but you wouldn't see any amyloid beta deposits in their brain. And then conversely, you will see a lot of amyloid beta deposits in the brain, but the severity of the Alzheimer's disease is not the same. It's actually significantly lower. And then the second problem is that all the drugs that target amyloid beta, either reducing their production or increasing their clearance, have failed over the last 20 years or so. In fact, the 2 types of FDA-approved drugs, there are 4 drugs for Alzheimer's disease are the ones which basically target glutamate receptors, which has something to do with calcium. They are basically calcium conducting channels, and they also target acetylcholine, which also is a calcium conducting pathway. So it looks like calcium has something to do with it. Khalid showed this slide. I just want to, he basically, just to remind you what he said was, there are these different scales of calcium activity, the ones which are happen at the single channel level or a few channels being clustered together. They have, some meaning for, for the signal function. They kind of have some instructions for the cell to do certain tasks. Then there are global signals which goes through the entire course, the entire cell, and then there are intercellular calcium signaling. So it's kind of calcium is a universal signal where these spatio-temporal scales of these signals, they have specific meanings for the cell function. These small ones, for example, they are important for mitochondrial function, like the energy production, vesicle secretion, the stuff that Suhita and Padmini talked about, synaptic plasticity, and the whole cell, they have fertilization, some of the stuff that Khalid talked about in gene transcription and so on. So if anything goes wrong at any of these levels with the calcium signaling, you can expect that the function of the cell and then the tissue is going to be disrupted. And that's why people floated this hypothesis, which is called the calcium hypothesis of Alzheimer's disease and neurodegeneration, is that actually it's the calcium which is causing a lot of problems in the brain in the aging diseases. And there is significant experimental in modeling literature on this hypothesis, supporting this hypothesis. So here are a couple of very influential views of that. So what are the questions that we are interested in? We basically, we use experiment is coming from Angelo. We have the NIH funding is with him, in collaboration with him. We do image and signal processing in modeling to basically answer two questions. What are the key pathways involved in the impairment of calcium signaling? And what are the primary downstream targets for the disrupted calcium signaling? So those are basically the two questions that we are after. So what is affected actually? I'll just, there are basically calcium signaling in the at the single cell level. It's affected by amyloid beta, which, remember, amyloid beta is the peptides, the protein that is related to the Alzheimer's disease. It's affected in different ways. One, if you expose the cell membrane to amyloid beta, it will make pores or channels in the membrane. And that pore can bring in calcium. And second, or actually another related, amyloid beta can also interact with calcium channels, other types of calcium channels, and that would bring in more calcium. The second way that amyloid beta can interact or affect calcium signaling is that if you inject in amyloid beta inside the cell, it activates the PLC pathway, which generates IP3 receptors, the stuff that Khalid talked about. And that IP3 would activate the calcium channel on the endoplasmic reticulum, releasing more calcium to the cytoplasm. Finally, the mutations in the proteins that are associated with the genetic type of Alzheimer's disease, presenilin, they can directly interact with the IP3 receptors. And they make these receptors to be more conductive or permeable to the calcium release from the ER. So those are the three things that we are trying to understand in relation to the first question that I posed. Okay, so but the end product is all this leads to higher than normal or toxic calcium concentration in the cytoplasm. That's just the end result of all this. Okay, so the first one is amyloid beta oligomers. They make calcium permeable pores in the plasma membrane. So if you do an experiment where you expose a cell or a cell membrane to the amyloid beta oligomers, and you wait for about 20 minutes, you will see that this amyloid beta will make pores in the plasma membrane. And if you apply a driving force, like, you change the membrane potential from 0 to somewhere -70 or -80 millivolt, you see these calcium fluxes coming into the cell. So the top part is basically the inside of the cell, and these puffs are these, these spikes that you see. That's calcium flux coming into the cell through these pores. And obviously you can expect that to affect the calcium signaling inside the cell. So the first question that we want to ask is, what are the functional properties of these pores? How they gate, how they function, how much calcium they can bring in, and all that. And then the second question is, do they evolve with time? Are they just, they are formed in the membrane and they stay, they have the same properties consistently for hours and so on. So those are the two questions that we would try to address with in relation to the pores. So first, to kind of give you the challenge, the experimental, the modeling and the image processing challenge here. When Angelo exposes, when he applies amyloid beta extracellularly, he waits. This is our, our experimental protocol. He waits for 20 minutes, and then he starts imaging because it takes the proteins about 20 minutes to insert themselves into the membrane and make these spores. So he would wait for 20 minutes, and then he would record the calcium flux going through these spores into the cytoplasm for about 30 seconds, which is about 15,000 to 20,000 frames a movie, which would have about 15 to 20,000 frames. And that movie would basically image about 20 to 40 micrometer wide region, 40 by 40 micron region of the cell membrane, and that has hundreds and thousands of pores. Then he waits for 5 minutes because you cannot expose the cell membrane to laser for too long. It causes damage. So he would wait for 5 minutes, and he does the same thing. He records another 30 minutes movie, wait for 5 minutes, another 30 minutes movie, and he keeps repeating that for as long as the cells survive. So there are these gaps basically where we don't have access to the to the information because the experiment is not on there. So that's one challenge. So the question is, what happens to the number of pores over time? Are they changing? What are the gating properties of individual pores at a given time? How does the calcium toxicity of pores change over time? So those are the things that we will try to address. Okay, so to make things worse, these pores are extremely small. They are basically a few nanometer size pores, and their open probability is really, really low. In other words, in a 30-second long video, if you zoom on one pore, that pore would open just a few times, sometimes even once for 2 milliseconds or 3 milliseconds, and then they disappear. And there are many of them, hundreds and thousands of them. So you cannot obviously look at them and detect them and characterize them just by hand, or you need automatic methods. And that's the first thing that we need to do. And then the second thing is you need to, if you want to track a given pore in multiple movies, now these multiple movies are recorded from the same cell and the same membrane. You just don't, you have this missing information because we are not recording for 5 minutes. We just record for 30 seconds and then wait for 5 minutes and then 30 seconds. So we need something, a way to track a single pore, how its properties are changing over time. And that's basically the first thing that we tried to do. So what we did was we developed this software, which we named Cell Specs. And it's a kind of really a few click software. It's a Java-based program, so you just open the program and load your movie from the experiment, and click a few buttons, and it will basically process your movie. And this is a map from about 40 by 40 micron membrane. The bright spots are the pores in the membrane, so it's their map. And then you can also, basically, you can just click on each pore and it will give you a time trace, which would change, which will, show you how the fluorescence or the calcium flux through these pores is behaving over time. So when the fluorescence goes up, that means the pore is open. When it goes down, it means the pore is closed. So just with one click, if there are thousands of pores, you can just save all these traces for each pore and then do whatever you want to do with it. Especially those are the things that are important for modeling. But so, again, this is just a location map from another cell. You can just pick on, any pore and look at the traces, the fluorescence change or the calcium flux through these pores over time. But you can also, this software will automatically generate statistics for all these pores in the movie. So for example, if you are interested on the, the distribution of open duration, the time for which the pores are open, it will give you that distribution, the time for which the pores are closed, the open probability, which is the relative time, so the time for which the pores are open divided by the total time of the experiment, and then the maximum flux, which is the amount of calcium coming in through these pores. You can basically get that information for each pore and then automatically generate these histograms. So going toward the modeling interest, another thing that we need for the modeling is, if you look at these traces, which comes from that previous software, there is a lot of noise in it. You basically have a lot of fluctuations. You don't even, you will see in the next slide, is that these pores, they have multiple conductance levels actually. So in one conductance level, they would be bringing in less calcium, and then the next one, they will be bringing more calcium and so on. So we need to remove this noise, and we need to idealize these traces where we could actually just look at the conductance level in which these pores are getting. And to do that, we developed this second software, which we call TraceSpecs. And again, you can load these fluorescence traces and just, click the button. And what it does, it would basically give you these nice clean traces where the step basically represents the conductance level in which the pores are opening. And then the zero, the flat line is the closed state. So if you look at these pores, they have actually very diverse properties. So for example, they have multiple conductance levels. In some of the cases, they will go up to 5 or 6 conductance levels. Some would just open to 1 conductance level, others would open up to 2 conductance levels. But what's interesting though is the conductance levels and the open probability increases over time, and I'll show some statistics on that. So when you first expose the cell to amyloid beta, these pores are extremely small, and they just bring in just a tiny bit of calcium, and they open very rarely. But then as the time goes on, their open probability increases and their conductance increases significantly. So here is an example of that. We look at 5 different pores. So in the beginning, which is, in the first movie, which is 20 minutes after we add amyloid beta, you see there is not much activity. But then as the time goes on in the second movie, in the third, and then by the time we reach the 7th movie, 60 seconds after adding amyloid beta, we see some of these pores have significantly higher activity. So that basically tells you that the toxicity of these pores is increasing over time. Another thing, as I mentioned in the beginning, we need to figure out that are these, when we go from, let's say, from movie 1, which I call stake 1 here, because this is actually, this image is we have collapsed the movie, which is 15,000 frames, into a single frame so that you could see just, everything in the movie. There are some pores which basically come from the first movie. For example, this is, movie 1 and 2 and 3. Some of the pores would be just on top of each other, meaning that they were generated sometime in the first movie, and they persist for the second movie and the third movie. But then there are some which are basically brand new. They are made in the second or third movie. And then you can do that with, as many movies as you like, for example, up to 7 movies. And then you do some, some statistics, some image processing to figure out that which ones are the common pores, meaning that, that are persistent throughout multiple movies or over longer time, and then which ones are the unique ones, which only appear in one movie and then they disappear somehow. But one thing that is really persistent is it doesn't matter if they are common or are unique. In other words, if they appear in a single movie or if they appear in multiple movies, over time, their toxicity, their mean open time, so for the duration for which they are open increases, the open probability is increasing, and then the maximum flux, or the flux through these pores, on average is increasing over time. And you can look at just the pore in the first movie, for example, and then in the second movie, the same pores in the second movie, and you will find out that the mean open time and the open probabilities are increasing. So the main message here is that the toxicity, the calcium, toxicity of these pores increases over time, almost persistently. So another kind of worrisome thing is that the membrane potential of the neuron and other cell types in the brain is close to this minus 80 millivolt. And unfortunately, at minus 80 millivolt, these pores are the most active. So when the cell is not functioning, they can bring in a lot of calcium, which could basically send false signals to the cell, or even consume a lot of ATP, because this calcium has to be recycled, and that requires energy. They are blocked by zinc. So if you apply A beta and then you add zinc in the solution, the majority of these pores would close. And the effect is reversible. So here, these are on the x-axis, you have time, and then on the y-axis, these are the pore numbers. So for example, here you have like 400 pores, up to 400 pores, and then the bright spots represent that the pore is open. So you see a lot of activity going on as a function of time, but when you apply zinc, the activity goes away. You wash out zinc, the activity comes back. The second pathway is when you inject amyloid beta into the cell, into the cytoplasm. What happens is it would activate the PLC pathway that generates IP3, and that brings in calcium to the cytoplasm. So these calcium ions, they can be localized. In other words, they wouldn't spread throughout the cell, and something like these clustered of these IP3 channels, they would release some amount of calcium, but that calcium doesn't spread throughout the cell. But then you also see situation where the wave or the calcium would spread throughout the entire cell. So you can see here the scale, it's 10 micrometers. So this whole width is about 35, 40 microns. So it spreads throughout the entire cell there. And then I guess this is kind of showing that the monomers, just the single peptide wouldn't cause any calcium release, but the oligomers, which are the where you have 7, 8, or 4, 5 monomers making oligomers, they actually are the ones which causes the calcium release. And as you increase the amyloid beta concentration, the amount of calcium release is basically almost linearly increasing. And these signals persist in the absence of extracellular calcium. So that basically means that the calcium is not coming in from outside. In this case, it's released from the endoplasmic reticulum.
Ghanim, which cells are these, by the way?
These are oocytes, okay. Xenopus oocytes, yeah. So, and just to kind of quickly mention that, this PLC-mediated IP3 generation and intracellular calcium release is not limited to Alzheimer's disease. So there are several, Angelo did these experiments a while ago. He applied amyloid beta. You see a big jump in the intracellular calcium. He applied prion. You guys probably have heard about the prion diseases. You see a lot of calcium, a significant calcium rise in the cytoplasm. IAPP is related to diabetes. Again, a lot of calcium released. Poly-Q related to Huntington disease and spinocerebellar ataxias, high calcium. And then lysozymes, which are related to immune system disorders. Again, the oligomers release a lot of calcium from the endoplasmic reticulum. The fibers, the long chains, they don't release any calcium, and the monomers, they don't release any calcium. It's just these medium-sized oligomers. And these, by the way, they don't stay permanently in the cell. They just appear for a few tens of minutes or a few hours, and then they disappear. They are dissolved and disappear. But those few tens of minutes or few hours is basically where they cause a lot of problems. So it's a very common mechanism for a lot of diseases, apparently. And then the third pathway that I mentioned in the beginning is that the genetic Alzheimer disease causing mutations in presenilin, what they do is they basically, these proteins are in the membrane of the endoplasmic reticulum. So they would interact directly with the IP3 receptor in such a way that the open probability of the IP3 receptor is significantly increased. So for example, if you look at the zero here at the single channel level, if you look at a single IP3 receptor, the channel in the wild type cells is mostly closed and it opens. One here represents open. It closes and opens, but most of the time it remains closed. The open probability, in fact, is somewhere close to 0.3, or it remains open for 30% of the time. And the model, basically, we develop a model for that which shows the same behavior. But if you take the cells with presenilin mutation, these channels remain open for most of the time, up to 82% of the time, which leads to a lot of calcium released into the cytoplasm. So again, a lot of calcium goes into the cytoplasm. And then it happens both in genetic and in age-related Alzheimer's disease. And in other
Ghanim, can you another 5 minutes maybe try and wrap up?
Yeah. So the question is, where does this higher calcium go? And the answer is, it's basically mitochondria is one of the main targets for the calcium. So when calcium is released, the cytosolic calcium goes up. Mitochondrial calcium, it basically goes into the mitochondria. It can affect ATP production, the energy production, in two different ways. It can increase the production of ATP through the TCA cycle, or it can decrease the production of ATP by depolarizing the mitochondria. And in case of Alzheimer's disease, the depolarization is more severe than the TCA stimulation. So cytosolic calcium goes up when you add different amounts of amyloid beta. The ER calcium obviously goes down because that's where the calcium is released from. The mitochondrial calcium goes up, and then the membrane potential of the mitochondria goes down, meaning the cell loses its membrane potential. And that leads to a drop in the ATP, and then a significant rise in the reactive oxygen species, which are also toxic. So we did some simulation basically where we look at the different variables as a function of amyloid beta concentration on the x-axis, and then a buffer AGTA, which would buffer calcium from the cytoplasm. So let's say when you increase the amyloid beta, the red color shows that the calcium in the cytoplasm increases, but then you start adding EGTA, and then by the time you add 3,000 or 3 millimolar EGTA, the calcium is completely buffered, so you don't see as much increase. And that you see that behavior in the mitochondria and the the other variables. But what's interesting is that the reactive oxygen species are also recovered, and the same with the drop in the membrane potential and the drop in the ATP is also recovered when we add AGTA. And of course, this is consistent with experimental results where if you add oligomers to the cells, a few hours, 40, 45 hours later, majority of the cells die. So you don't have any cells remaining after 40 hours. If you add AGTA, which is shown by the red curve here, the cell viability recovers. Lithium and PTX would block the IP3 generation. So if you block the IP3 generation, no calcium would be released from the ER, and that also helps the cells to restore its viability. So just to summarize what I discussed, a beta oligomers form pores in the membrane with a wide range of gating properties. They become more toxic over time. Zinc can block these pores reversibly. Intracellular A beta oligomers stimulate IP3 production through PLC pathway, which causes the release of calcium from the intracellular stores. And the genetic type of Alzheimer's disease, the mutations, presenidine, they cause calcium release from the intracellular store by upregulating the function of IP3 receptors. High cytosolic calcium leads to hyperexcitability, mitochondrial dysfunction, and fragmentation and impaired synaptic function. So these last points, the hyperexcitability and the mitochondrial fragmentation and impaired synaptic functions, we have done that modeling and we have the results. There are a few papers that are in preparation, but I don't have time to go over that. So hopefully maybe in some other time. But that's all I wanted to say, and I'll open it to the questions.
Okay, good, Ghanim. Yeah, no, this is very actually. I need to sort of talk to you about your software, because I think well, of course you've used it for what you're interested in, but there are other I can see a lot of other potential uses. We have, so I'm sorry to be kind of strict here on the time, but we have two sessions coming up. One is where it's really Linda Kenny and Mike Sheets, who founded the Mechanobiology Institute in Singapore, and they have given us, they couldn't be present personally just because of the timing, but there's a very nice 40-minute presentation, a movie that they have given us. And then I will need to introduce that movie, and I will need to also try and put the bits and pieces together. So I'll try and take 10 minutes or so. So the movie is 40 minutes. My thing would be 10, 50 minutes, and then a few minutes for questions. So that session starts at 12. Maybe just a quick couple of questions. What I would suggest, there won't be much time for, we already have a fairly extensive question and answer after Khalid's talk, which I think was great. But I would really recommend people to put their question in the chat box, because if you do that, we will get to it. I mean, we won't do it, immediately, but we will get to it. And in fact, so, if I could please encourage, please do not, think of questions, put them in, and we'll get to it. Okay. Now, actually, there's one question we can even take because there's a little bit of time here. Let's, Suhita, you had a question?
A quick technical question.
Yeah, why don't you ask that?
Your model, does it include depletion and then the activity of SOCs? That's question one. Question two is, again, there are these three very distinct ways in which calcium goes up, right? There's sort of over signaling. Which do you think is there sort of an overwhelming one, or have you tried putting all three in the model to see which has the drastic impact? Because at some level they're competing for the same calcium, right? So one pathway must take over the other, or how do you think about these things?
So, so the question actually, the answer to both questions is no. So we haven't actually taken the depletion and the store operated calcium entry into account at this point. We just basically finished the three separate components. So we have two papers that are going to be submitted hopefully in the next month or two on just the A-beta modeling and analysis, and also their evolution. We published the IP3 receptor sensitization part already, so that has been done. We also published the IP3 receptor, the IP3 stimulation by the intracellular beta, but we haven't put all them together. So we are basically going in that direction, but it's kind of just building the individual pieces is taking some time. So hopefully maybe in the next couple of years, maybe hopefully sooner, but
I think that's what we are there for, right? Modelers, we can actually put these three things together in a single system and figure out the power of modeling.
Khalid, did you have a quick question? Yeah.
Quick question, very quick.
Yeah.
I'm wondering, Ghanim, looking at the A-beta channels, whether they're actually pores the way we think about typical channels, or whether they're just hydrophobic molecules inserting into the lipid bilayer and just generating calcium influx, especially at longer time points when you see this, basically everything break loose and you have a humongous flow of calcium.
Yeah, so actually that's a good question. And we usually, the reason, because of your comment, we usually don't use the word channel for them. We call them pores because they do seem to have channel-like properties. They basically open and close. They have multiple conductance levels. So they are not, kind of pores where you apply a driving force and the calcium just keeps flowing in. They would open and close, and they are, all of them do that. So that's something that is, there is a gating mechanism to it. We don't know it, where that gating mechanism comes from. But they are not just openings that, that would just allow a non-stop flow of calcium.
Yeah, but, so they're not pores. The pores are discrete states, I guess.
Yeah, we like I said, the reason we use the word pores for them, because the exact same thing that, that Khalid is pointing out is, these things come in, how do we know these are channels? Because channels are supposed to have ligands or voltage dependence. These pores do have voltage dependence, but it's just the dependence is coming through from the driving force. It's not like the gating mechanism.
But that's good enough for the coarse-grained level of your simulations. I mean, that's pretty much all you need to define it.
Yeah.
So okay, let's take a